Hello, this is Ed Chapman, and this video cast is going to explore the structure and function of the endoplasmic reticulum, vesicles, and Golgi bodies. Now, the endoplasmic reticulum, otherwise known as the ER, and completely feel free to use ER from now on, that's a lot to write, is built from a much folded and stacked up assembly of very flat sacs that kind of form a maze or a membrane throughout the cytoplasm. The ER comes in two forms. There is the smooth ER and the rough ER. The ER is centered around the nucleus, which means there's a lot of endoplasmic reticulum surrounding the nucleus and then extending from the nucleus outward into the cytoplasm. The ER has an inside space that's separate from the cytoplasm itself, and the function of the ER is to build and transport some different materials depending on what kind of ER you're talking about. Now, the smooth ER is smooth because it doesn't have ribosomes attached. And remember, ribosomes are protein factories. So the smooth ER really doesn't do anything with proteins. What it does is it builds and stores lipids and carbohydrates. All right? So the smooth ER is involved in the metabolism of lipids and carbohydrates. Okay, the rough ER, because it has ribosomes attached, is taking the proteins that are built by the ribosomes and storing them and perhaps changing them in some way. All right, so you can think of the rough ER as the storage and building location of a lot of proteins, but it's only because it has ribosomes attached. Okay, now it's very important to remember that the smooth ER is involved in lipids and carbohydrate metabolism, and the rough ER is involved in the storing and metabolizing of proteins. All right. Now here's a picture or a diagram of the ER, and you'll notice the two different types. This part here that's colored kind of orange is the smooth ER, and you'll notice it kind of blends into the rough ER. The rough ER has the ribosomes attached to it. All right, and notice here's the nucleus. So that's what I meant by the ER being centered around the nucleus. Here's an electron microscope picture of some real ER, and I can't really tell if it's smooth or rough, but you can see how it's just really extensive. There's a lot of it spread throughout the cytoplasm of the cell. Now, I like to think of the endoplasmic reticulum as a highway transport storage system all kind of combined together. Because it weaves all through and among the other organelles in the cell, that's why it makes me think of a roadway or a highway system. Now, the Golgi bodies are a little bit different. They are more compact, but they share a lot of features with the ER. They're made of this folded stack of sacs, but they're arranged in layers rather than like a maze or a highway system. And the function of the Golgi bodies are to, let me go back here. The function of the Golgi bodies is to assemble and package complicated molecules for transport out of the cell. That's probably the most important thing to remember here, for transport out of the cell. So what the Golgi does is it takes material and puts it in packaging that allows it to leave the cell. So let me show you what I mean there. This is an actual picture of Golgi, and you can see the flattened layers here, okay, right here. And coming out of the flattened layers of these sacs called vesicles, right? And they're pretty dramatic in this picture here. This kind of lava lamp kind of structure, all right? And let's see how that works. So a vesicle is just a sac of something. It's surrounded by membrane and it's got stuff on the inside. And what the Golgi apparatus or the Golgi body is able to do, sometimes it's called, let me go ahead and give that to you guys. Sometimes the Golgi bodies are called Golgi apparatus. Some books use that. Okay, I'm honestly not sure which one your book uses, but it's the same thing, same idea. But what the Golgi bodies are able to do is take in vesicles of material, okay, in their top layers, and as the material kind of bubbles and oozes from layer to layer to layer, it finally is inside of a package at the end, okay, so it's surrounded by a membrane now that allows it to be exported out of the cell. All right. So what you get at the end of this process is some cell product that's ready for export. And cells produce lots of different products. You probably are familiar with some of these. For example, there are cells in your salivary glands that export saliva. And there are cells in testes and in ovaries that export two of the hormones that control the development of different sexual characteristics. You probably have heard of testosterone and estrogen. Analogy-wise, I like to think of the Golgi apparatus or the Golgi bodies as assembly line factories. They're building and packaging products for shipment outside of the cell. In the same way that in this picture we see workers assembling iPhones, and these iPhones are being packaged in this truck here to be shipped out of the factory. So the Golgi bodies kind of do a similar operation for the cell. All right, we'll stop there. Thanks for listening.